Okay, I think we're ready to start. I'd like to welcome everybody to Pediatric Grand Rounds. Today's a special Grand Rounds, and I'm going to hand it off to Dr. Fernando Gonzalez to introduce our speaker today and to explain a little bit about why this is a special Grand Round. So, Fernando.
Okay, thank you, Dr. Hirsch. So, yes, today is our second physician scientist interview day. So today it's my privilege to introduce as our Grand Round speaker, Dr. Alejandro Sweet-Cordero. So Alejandro obtained his undergraduate degree at Stanford, came to UCSF for medical school and his pediatrics residency before going to Boston and the Dana-Farber Cancer Institute for his subspecialty training in pediatric hematology oncology. He was at Stanford for a number of years before being recruited back to UCSF in 2017. He'll talk a little bit about his background. His expertise is in cancer biology, genomics, and translational preclinical therapeutics. So please welcome Dr. Alejandro Sweet-Cordero.
Thank you to all the organizers for the opportunity to speak to you today. Do you see my slides? Am I good?
Yes.
Okay, great. So thanks, Fernando, for the introduction. The title of my talk today is Precision Medicine for Advanced Pediatric Cancers, the UCSF experience. I was asked to just because this is a special Grand Rounds in honor of the physician scientist interview day, to talk a little bit about my career trajectory. So apologize for indulging in my own career here a little bit. But as Fernando mentioned, that I was a undergraduate at Stanford. I double majored in biology and anthropology, and the only relevance to that, I think, now is that I think actually having majored in anthropology does give me some insight into the interesting issues of cultural diversity that we have to deal with on a day-to-day basis on the clinical side, and I think it's helped me perhaps be a little bit more in tune to that. I got an early start in research working in Richard Scheller's lab. He was very supportive of me early on, which I've appreciated. And Richard then went on to be the CSO at Genentech. So he's been extremely successful. Then I came here to UCSF to do my MD. I had several great mentors, including Chip Chambers, who was at San Francisco General, Jose Santos, who's a mentor and a good friend from the Hospital Infantil in Mexico City. I did my residency here, and that's where I kind of started to think about oncology as a future career. I was really supported very significantly by Kevin Shannon and Kate Mette, who were here at the time and continue to be here and really encouraged me to pursue my passion to link together my work as an oncologist and work as a scientist in the area of cancer biology. Then I worked as a clinical instructor at San Francisco General for a year with Kevin Coulter before moving on to the Dana-Farber, where I did my fellowship training, also at MIT with Tyler Jacks. I got great mentorship from him. Also worked with Todd Golub. And I mentioned these names really just to emphasize to all of you who are young trainees the importance and the value of mentorship. And mentorship is a kind of a two-way street. It's really a relationship, just like any other relationship you might have in your life. You need to work on that relationship. You need to kind of keep in touch with those people, let them know what you've done, where you're going. And I can tell you that the people that I've mentored, I always really enjoy hearing from them, even if it's been many years since I worked with them directly. So I would encourage you to think of these as relationships, a little bit like a plant. If you sort of just put it on your windowsill and don't water it, it's going to kind of wilt away. But if you keep watering it and keeping in touch with those people, it's sort of a gift that keeps on giving. So then I was at Stanford, as Fernando mentioned, and was very happy to be recruited back to UCSF. And I've been here happy as a clam since 2017. So I think it's a great environment for physician scientists in particular to pursue the sort of the interface between what we like to do in terms of taking care of patients and really driving cutting edge science. And so those of you who are considering applying here, I really can't think of a better place, having been at many other top-notch institutions. I think this is the top playground for physician scientists. So I would encourage you to think about coming here. So my lab is at Rock Hall here in the Mission Bay campus. This is one of these do as I say, not as I do. Everybody always talks about being focused. I've been probably a little unfocused in my career. We work on a lot of different things. We work on lung cancer genomics, particularly as it relates to oncogenic KRAS and tumor heterogeneity and lung response, and therapy response. We also work on, in the lower half of the slide is our emphasis on sarcoma biology, but I'm really going to focus on the middle lower side here with angle, which is this real world application of genomics to advanced pediatric and adult young adult cancers. Probably not going to have a chance to talk about our work in osteosarcoma. So the outline for the talk is really to first start talking about our efforts to advance precision cancer medicine in using this test that we'll describe called the UCSF500, and then talk a little bit about our efforts to bring in more cutting-edge genomic technologies to advance the care of patients who have relapsed in our clinical setting. So what is our vision for pediatric cancer genomics? So this is a little figure that we put together for review, published a couple of years ago. We encounter patients kind of in two settings. One is a diagnosis, as you see here on the left, and then unfortunately a fair number of our patients actually relapse. And so both a diagnosis and a relapse, we feel strongly that developing a comprehensive workflow that allows us to look at all of these different elements shown on the pie on the left at diagnosis, and also evaluate the germline of the patient, but then also when patients relapse, being able to reevaluate the tumor to understand how the tumor evolved, what made it relapse, what made it different. Hopefully, gathering all this information and learning new ways to analyze the kind of the flow of information that we get from these different sequencing technologies will help us identify better therapies for our patients. So when we talk about precision cancer medicine, maybe we should start with some definitions. Sometimes people also call it personalized cancer medicine. I don't like the term personalized so much because it implies that it's going to be sort of unique for every patient. But I think it's going to be probably more like groups of patients that are going to receive similar approaches. But the idea is to tailor the therapy based on the genetic analysis of the tumor itself, rather than our traditional approach in oncology, which has been to focus on the histology or the classification of the tumor. And the goal is really to match the right drug to the right patient, hopefully to increase response and to decrease toxicity. Here at UCSF, we've implemented this through an initiative through the Cancer Center led by Alan Ashworth called the Molecular Oncology Initiative. And we have something called the Molecular Tumor Board, which I'll mention in a little bit. So in pediatric oncology currently, I should say that many clinical trials already incorporate genomic biomarkers based on sequencing or some type of molecular analysis. And the idea is to assign patients based on the presence of these specific molecular markers. But that idea that you would assign patients based on the biomarker, except in some diseases like leukemia, is still relatively untested. So many early studies have now been published, and they focus primarily on feasibility. There's a large study underway called the Pediatric Match, which is looking at this idea of matching specific molecular biomarkers to targeted therapies. One limitation of the Pediatric Match, which is being kind of addressed in the next iteration of it, is that it only looked at individual drugs. And we all know that in order to cure cancers, we need combination therapies and not just individual therapies. One thing that has happened in the last few years, and actually just started this in the fall, is the implementation of something called the Research to Accelerate Cures and Equity Act, called the RACE Act, which is a law that was actually passed by Congress and started this last September, that requires that all drug companies develop a pediatric development plan, which didn't used to be the case. There was no sort of incentive for them to do that, but now they're sort of required, which has really increased. We've seen it happen in my own lab, the amount of interaction we get now from pharmaceutical companies reaching out to us, wanting to develop a plan for specific pediatric cancers, that should really increase the number of sort of gene-drug pairs that we have to be able to test in these kinds of more personalized approaches. So here at UCSF, we've developed this assay called the UCSF500, which is a next generation sequencing assay that evaluates both the germline and the tumor. So you can draw blood to evaluate the germline and then obtain a biopsy and then sequence both of those. And then here you see depicted the idea that this is a panel assay. So you're basically selecting probes from the germline and from the tumor, and you're sequencing only those regions of the genome that are covered by those probes. So it's what we call a panel assay, as opposed to what I'm going to talk about later, which is whole genome sequencing. So this is a test that was developed here at UCSF in consultation with both adults, and I put "and" in bold because one of the really nice features of it, and one of the nice features of UCSF, since I'm trying to do a little advertising here, is that as opposed to a lot of institutions where pediatrics may be kind of siloed or separated from the adult efforts, we have a very strong integrated program here. The fact that I'm the director of the Molecular Oncology Initiative, which is 90% adult cancers, kind of speaks, I think, to the respect that the pediatric community has at UCSF, and the fact that we aren't siloed in any way. And we were very central to the development of this assay, so that makes it particularly relevant to our patients. The DNA, this is a DNA-based assay. The DNA is extracted from formalin-fixed tissue for the tumor, or blood, or sometimes saliva for the germline. Currently, a limitation is that it's a DNA-only test. It doesn't cover, doesn't evaluate RNA. We'll talk about why that's an important limitation later. So as I mentioned, I'm the director of this program called the Molecular Oncology Initiative. It's run out of the Cancer Center. The goal of this is to serve patients and providers by providing state-of-the-art evidence-based recommendations to help guide interpretation of molecular testing performed on cancer patients at UCSF. And we do that through a molecular tumor board, which I'd encourage you to join if you're interested in this topic. We meet twice a month, and if you email me, I can provide you with more details. So we also develop methods for measuring utility and feasibility of precision medicine in all of our patients, with the goal of learning from current patients to help future patients. And I'll talk a little bit about that as well. A very important component of what we do is also developing platforms and methods for data sharing and innovation. You're not going to be able to make much progress if the information for each individual patient is isolated and you can't kind of pull it together. So much of what I'm going to show you today is through these data sharing efforts that we've developed here at UCSF. So back to the test itself, it's as I said, it's a panel assay that covers about 479 cancer-relevant genes, so the exons for those genes. It also tiles through the introns for about 47 genes to facilitate fusion detection. We also have probes across the genome to look at copy number. It sequences to a depth of 500x. And I should say that the test itself is something that's done in a CLIA lab here at UCSF. I have nothing directly to do with the test itself. It's run through the CCGL lab, which is a CLIA certified lab, where molecular pathologists do the interpretation. They use a platform called DNA Nexus. The turnaround time is about 14 days. That turns out to be important, obviously, if you want to be able to use this information to impact on patient care. And then the clinical report is generated, and so we take over downstream of the clinical report in terms of the molecular tumor board. This is just an example of what the report looks like. This is actually for an adult patient with a solid tumor adenocarcinoma in the liver. But the point I wanted to make here is that you can see down below in gray, these are pathogenic alterations found in the germline. So this particular patient has a BRCA2 mutation in the germline that was identified because the germline DNA was sent. Interestingly, there's a second hit in the second allele of the same gene, BRCA2, which is a strong indication that this is likely not just a incidental finding in the germline, but something that is pathogenic to the development of this particular tumor. There are also alterations shown in the blue arrows in other genes, in this case, single nucleotide variants that are likely important for the pathogenesis of this cancer. So in general, the utility of these kinds of assays is threefold. One, it can identify cancer risk, such as this case. So obviously, this particular patient had a tumor that was initiated by a germline loss of one allele of BRCA2, which is important for treatment of this particular cancer, but assuming that we can cure this cancer will also be relevant to risk assessment further on for the lifetime of this patient in terms of following them and screening them for development of secondary malignancies. In the red arrow, we see the detection of therapeutic targets, in this case BRCA2. It turns out that patients that have loss of BRCA2 are susceptible to treatment with something called a PARP inhibitor. So this report actually identifies a potential druggable alteration in this patient. And then lastly, not relevant to this particular case, but oftentimes, I mean, perhaps surprising, we tend to think that, oh, we know how to diagnose cancer, but particularly in a referral center like UCSF, we see a fair number of cases where the diagnosis is actually in question. And even with our, stellar pathologists, often standard approaches don't really give us the diagnosis. And the molecular assay can sometimes really clinch the diagnosis and help the patient, help us find the best therapy for that patient. So I'm going to spend a little time just going through the kind of our experience of the cases that we've sequenced here at UCSF, because I think there are some kind of interesting aspects to it. I should say that much of this work is facilitated by a really strong team of computational biologists, Courtney Onodera, who's part of the CCGL team, Carlos Espinosa, who's part of the Molecular Oncology Initiative team, and Henry Martel, who made many of these figures, who is a postdoc in my lab. And so this data just summarizes the experience here, sequencing cases using this UCSF500 assay. It's kind of a complicated figure, but if you look at the left, you'll see the kind of common pediatric cancer diagnoses. CNS, obviously brain tumors, carcinomas, we don't see very many, but we do see some. Non-rhabdo soft tissue sarcomas, rhabdomyosarcoma, osteosarcoma, Ewing's, and I won't get into all the specific diagnoses, but the point being that we've sequenced cases across a large number of diagnoses and across a large swath of different ages, so 0 to 9, 10 to 18, and then AYA, which is 19 to 30. We've been we have a very strong cohort of brain tumors because the brain tumor group was an early adopter of our assay. We've also been able to, in many cases, do tumor normal, which is what we prefer to do, but for a variety of reasons, oftentimes only the tumor gets sequenced and the normal does not get sequenced, and we've tabulated that here. And then importantly, on the right side here, you see the stage at which the tumor was sequenced. A very large number of cases, the ones shown in kind of brown and purple, are sequenced from metastasis. And so obviously we like to sequence the case closest to the, when we're going to actually treat. Sometimes we can't do that. We're reliant on a sample from the biopsy, but as much as we can, we try to sequence the metastatic samples. And so this is an incredibly rich data set that I'm only going to scratch the surface of today. Those of you who are interested in any aspect of precision medicine, it could be, understanding cancer predisposition. It can be understanding the role of particular genes in cancer. We also have people looking at this data in terms of understanding aspects of disparity. So Dr. Weinstone in our division of bone marrow transplant is quite interested in understanding, who gets this sequencing. Are there biases in terms of socioeconomic condition, or potentially ethnic or racial disparities, and those can all be evaluated here as well. So it's an incredibly rich data source, which for any aspect of research in our institution. So we've sequenced a total of almost 1,200 cases now. I said about 2/3 are brain tumors because, as I mentioned, they were early adopters. Although I will say that now, through something called the Destination Program, which I'm going to mention in a few minutes, we are sequencing pretty much every patient that comes through the pediatric clinic with a malignancy. So these numbers are going up quite rapidly. And again, this will be a very rich resource for many investigators. So this figure, these figures, I will say that one of the more difficult parts of doing this kind of research is how to visualize this data. There's a lot of information. How do you visualize it in a way that, you know, the human eye can kind of understand? And I won't say that we've achieved that, you know, to the degree that we would like to, but we know we're working hard to find ways to visualize this information to make it useful. So here you can see, again, by diagnoses, the type of alteration that we see in the UCSF500. The different colors indicate whether it's a copy number variation in the green colors, whether it's an insertion or deletion, a fusion. So for example, in Ewing sarcomas, they all characteristically have the EWS fly alteration. So that's why, you know, that's a significant fraction of the total number of alterations seen in these tumors, whereas fusions are less common in other diseases. But we also find some kind of novelty here. So for example, I wasn't aware that in a large fraction of our hepatoblastomas, insertions and deletions are quite common. And so, you know, the more you stare at these plots, the kind of more things you learn. And diving into kind of more specific genes that are altered, perhaps not surprising to any of you who either study cancer or remember your cancer class from undergrad or medical school. P53 is a tumor suppressor that is widely responsible as a driver for many cancers, and it's the most most common alteration that we see across our cohort. But interestingly, you know, it varies in terms of how common it is across different diseases. Almost never seen in hepatoblastoma or neuroblastoma, very common in osteosarcoma. So you can start to pick out some kind of both known things that that are well known in the literature, but also some novelty here in terms of things that we wouldn't necessarily expect. So beta-catenin is very commonly mutated in hepatoblastoma. That's well known. Unfortunately, one point to make here is that these are the most common alterations that we see across our cohort. And one of the big limitations, or perhaps the biggest limitation to drive precision cancer medicine in pediatrics, is that the most common alterations continue to be undruggable. So pretty much every one of these alterations that I'm showing you here, we do not yet have a drug that we can specifically pull out of a drawer and give to a patient because they have a beta-catenin mutation or because they have a MYC alteration. That's beginning to change. So now, for example, KRAS, which for decades has been considered to be undruggable, there's a subset of patients with KRAS mutations that now have a druggable alteration, but that's still relatively uncommon. We also pick up a lot of germline alterations. So this is the looking, so summarizing the germline data from our cohort of over 1,200 cases. Again, P53 is the most common alteration that we see in the germline, and that's what we call the Li-Fraumeni syndrome, which is caused by P53 and a few other genes that are the cause for specific subsets of pediatric cancers, primarily osteosarcoma, some subsets of leukemias and other sarcomas, as well as other cancers that are known to be driven by loss of P53. So we see that, again, that's not particularly novel, but we do see some more unusual alterations, like for example, if you look at the bottom, ASXL1, as far as we know, has not been previously associated with hepatoblastoma, and we see that as a significant cause in some of our cases. The most important take-home message, I think, for not just for oncologists, but for general pediatricians and the community at large, is that perhaps not surprising to many people, is that a very large fraction in our cohort, 17% of all cancer cases seen have a genetic predisposition. And so this is something that if you looked at in a textbook of pediatric oncology published 5 or 10 years ago, they would have said, oh, somewhere between 3 and 5% of cases are caused by genetic predisposition. And now through our sequencing efforts, but also published work from the group of Sharon Plan at Baylor and the group at St. Jude's, we know that that's actually not true, and that the genetic component of cancer is quite high, can be as high as 20 or 30% in some malignancies, as shown in this next slide. So for example, in osteosarcoma, over 30% of the cases we've diagnosed so far have a genetic, or that we've sequenced so far, have a genetic predisposition. Much less common, say, for example, in Ewing's or in neuroblastoma, but not not unseen. And looking at the germline can also help us to potentially identify some novel mechanistic insights. So this is a little paper we published last year where we looked at a case of a patient with Ewing sarcoma who also happened to have neurofibromatosis type 1, which is a genetic predisposition that has not been in the literature very strongly associated with Ewing sarcoma. But what was interesting was that we saw actually loss of the second allele in the tumor, suggesting that there was a selective pressure for the tumor to lose the second allele. Loss of NF1 is one way to activate the RAS pathway. And there's been some kind of suggestions in the literature over the years that maybe there was a connection between Ewing sarcoma and RAS. And so this paper, which is just a case report, kind of helps to strengthen that notion. And I think we find a lot of these kind of one-off sort of interesting molecular case studies. There's actually a journal now called Molecular Case Studies where you can publish these. And so again, for those of you who are residents or medical students or interested in oncology, this is a great way to get involved in working up one of these cases and thinking about what it might mean for advancing our field. So in terms of actionability, what can we actually do with all this information? So this kind of summarizes what we call, we use something called OncoKB, which is just one way to kind of characterize levels of actionability. It's a widely utilized classification. So level 1, shown in green, is what's is FDA approved for that indication. So the FDA has agreed that this drug works for this disease when you have this molecular alteration. Level 2A is when there's a match to the gene, but not to that drug in that disease. So, for example, it's a drug that's been approved for osteosarcoma, but you're trying to use it for a patient with Ewing sarcoma. That would be a 2A, and so forth. So, level 3A and 3B then is there's some clinical evidence in that disease, but it's not been sort of FDA approved. And 3B is that there's clinical evidence in another disease. And level 4 is when there's preclinical evidence from experiments in the lab, but it hasn't been taken to the clinic yet. So unfortunately, the first thing you'll notice when you look at this is that many of our diseases still are in gray, indicating that there's no recommendation, as I showed you earlier, or level 4, so basically something in black. So there's some indication that the EWS5 fusion can be targeted, but that's not been sort of taken to the clinic yet. And very few times do we actually see something that is a kind of a level 1. And that again speaks to the historically to the lack of specific development of drugs for pediatrics, and it's because of the RACE Act that's beginning to change, but still lots of work that we need to do there. I will say that one of the advantages of the UCSF500 is it allows us to identify fusions because, as I said, it tiles across the introns, and we find oftentimes fusions that we would expect, like EWS. So the way to read this is the N-terminal fusion is shown on the y-axis, and the C-terminal fusion is shown on the x-axis. So here you see EWS fly, which would be sort of the most common number in terms of how commonly we see it. We also see other EWS partnered fusions, for example, EWS ATF1, EWS BEN2, EWS WT1. The ones that have an X are ones that are kind of canonical, meaning that they're sort of recognized in the oncology community as being, well-known fusions. And that's mostly what we find because that's what we're looking for, because again, we're tiling across these introns. But occasionally we do see fusions that we wouldn't have expected. We pick up sort of incidentally. And some of these probably are actually quite important pathogenically for these patients, and we'll talk a little bit more about those later. We're limited in our ability to detect fusions, however, because we don't have yet in the CLIA certified CCGL lab an RNA-based assay, but they're working hard to develop one. So one of the things that we've tried to do in the lab with this large data set is think a little bit more about what we call second order genomic alterations. So what I showed you already is the single nucleotide variations, in both the germline and in the tumors, the fusions, things like that. But there are kind of broader things that you can learn from these DNA sequencing panels that can be quite relevant for patients. So one of them is the overall tumor mutational burden. So how many mutations do you see overall if you look at the entire footprint of the panel that you're looking at? And that's been studied quite extensively, primarily in adult cancers, as potentially a way to identify patients who would be more likely to respond to immunotherapy. So as you probably are aware, there's been a huge revolution in pediatric oncology over the last 5 or 6 years in terms of our recognition that we don't need to just target the tumor cells themselves, but if we can kind of reawaken the immune system to recognize tumors, that kind of dramatically increases our armamentarium. And you've probably heard of these kind of famous cases, Jimmy Carter living with a disease that would have killed him 10 years ago, metastatic melanoma. He was essentially cured, or we think he's cured with immunotherapy. That has been less effective in most pediatric cancers, with the exception of CAR T-cell therapy and some forms of immunotherapy in liquid tumors, less so in solid tumors, but nevertheless, there's this potential for tumor mutational burden to impact on some of our patients in terms of identifying those that might respond to immunotherapy. High tumor mutational burden can also identify patients who have specific genetic risk factors, for example, something called a mismatch repair deficiency, and also identify particular types of responses to prior therapy. So, for example, radiation and chemotherapy. I'll talk a little bit more about that later. Lastly, analyzing mutational burden, more specifically looking at the types of mutations that you see using an approach called mutational signature detection, can also help us to identify potential environmental causes or potentially intrinsic mutational processes that are relevant to specific cancers. So here's a kind of a summary. I should say that we're limited in our ability to look at tumor mutational burden and particularly mutational signatures in the panel data because it's a specific footprint. It only covers about 2.5 megabases of the genome. So unless you have a specific number of mutations, we're kind of limited there. So, but we do see a subset of patients who have what we call ultra hypermutated cancers. There's about 11 of those, and then hypermutated cancers, and those are the ones that we think might be more likely to respond to immunotherapy. And some of these patients have already received immunotherapy, and how effective that's going to turn out to be in terms of linking it to tumor mutational burden in pediatrics is still something that's an open question. This is just another way to look at the same data. How many patients we see that are ultra hypermutated is a very small fraction, even the hypermutated patients. There's this other group that in pediatrics we call pediatric high, meaning that they have a higher tumor mutational burden than the average pediatric patient, but whether they would be more likely to respond to immunotherapy is still something that we don't know, and is actively being studied at the moment. I mentioned these mutational signatures. I won't get into this with too much detail, but the idea basically is that you don't just have to look at the specific mutation itself. In this case, if you look at the left here, signature 6, a C to A or a C to G, but you can also look at the context in which that C to A or C to G alteration occurs, and the context in this case being the 5' and the 3' nucleotides to either side of that mutation. And so if you put all those combinations together, you end up with 96 possibilities. And so, for example, this signature 6 is a signature characterized by C to T translocations, where there's a kind of a specific pattern of frequency of the 5' and the 3' alterations. And so that's what we call signature 6. There's another signature called signature 3, which is kind of more flat. Signature 2, which is a very specific spike in the C to T here. So there's a total of about 30 of these mutational signatures, and they can tell you quite a bit about both what are the kind of pre-existing exposures that this patient was, or the tissues in this patient were exposed to, and also kind of the evolution of this tumor. So we can look at these mutational signatures in a subset only of about 60 cases. The ones that I mentioned to you earlier were either hypermutated or ultra hypermutated, and that's because we need at least 50 mutations across this panel in order to be able to distinguish what the mutational signature is. But again, you start to see some kind of interesting patterns here. So I'll mention a little bit later something called the APOBEC signature, which we see in a subset of our ALL patients, deficient mismatch repair, deficient homologous repair in other hematologic malignancies. And some of these can be, for example, we see evidence of UV exposure and some of the other pediatric cancers. So again, this can tell us something about the etiology of these cancers, and it's something that we're also quite interested in looking at. Just to kind of wrap up this first part of the talk, we've put together all this information that I mentioned to you in something called the UCSF CBio portal. Anybody at UCSF can access this information through My Access. If you go to My Access and you look at UCSF CBio portal, you can request access and you'll get access to a platform that looks something like this on the left. For example, if you were interested in understanding subsets of brain tumors, you could identify how many anaplastic pleomorphic xanthorostenomas we've seen here at UCSF, and the answer is 25, or any other specific subset here. There's a huge amount of information. We're working very closely with the Institute for Computational Health Sciences to enrich this information with phenotypic information from the medical record, including survival, therapies received, response to those therapies using imaging analysis, and many other, ethnicity, for example, many other parameters that could be interesting to a variety of researchers looking at many different aspects of precision cancer medicine. As I mentioned briefly, we have a program called the Pediatric Precision Cancer Medicine Program, which is a clinical effort to kind of bring this, all of this knowledge and all of this information really to bear to help our patients in real time and in real life. So we get this UCSF500 test either from outside referrals or from our own patients. We discuss it at our tumor board that can then generate information that can be used to, for example, enroll patients onto specific phase 1 clinical trials, or perhaps help the local oncologist taking care of this patient to better think about how to track this patient if they have, for example, a germline predisposition and so forth. This program started in January of 2020, kind of had a slow, painful start because we got stuck in the middle of the COVID pandemic, but we're hoping to relaunch it in early 2021 with focused energy. So just to kind of wrap up what we can learn from these DNA panels, they can clarify the diagnosis, they can help us identify germline predispositions, rarely identify actionable single nucleotide variants. But one of the most frustrating aspects of this, as I mentioned earlier, in addition to the fact that many of the things we do find are not actionable, is we see a very large number of patients who, for example, have very advanced cancer, metastasis in the brain, metastasis in multiple organs. We biopsy those tumors, we send them to UCSF500, and the report comes back with nothing, literally nothing. So of these 437 exons that we sequenced, the 47 introns that we sequenced, and the copy number, we don't see any alterations. So we think of this as the kind of the missing signal in pediatric cancer. So what is the driving these cancers that we don't capture in these kind of reduced panel assays? So are there structural variants that are driving enhancers to increase gene expression? Are there non-coding mutations? Are there other epigenetic alterations, rare fusion events? And so thinking about that, 4 or 5 years ago, we launched this kind of integrative genomic effort first at Stanford, and then we moved it here when we came to UCSF. And so the idea is to really try to, working very intensively with our surgical colleagues, with our pathology colleagues, with a team that we've assembled here in the lab, to try to obtain as many of these both primary and metastatic samples as we can, do not only the panel testing, but also whole genome sequencing, RNA sequencing, as depicted on the left. And then we have a very active effort to develop what we call patient-derived xenografts, which are basically tumors that are implanted directly into mice, so that we can expand these tumors and have a material to work with, both to generate cell lines, to sequence the expanded tumors, and to, for example, develop preclinical trials to test novel therapies in the mice that would be very difficult to test in patients, particularly for these very rare diseases that we only see maybe 1 or 2 a year. So again, this is kind of an overview of this integrative genomics workflow. We obtain tumors at diagnosis and at relapse, both germline and somatic tumor tissue. And then we can apply all these various technologies and methodologies that are depicted here. We can evaluate the germline deeply. We can look at somatic DNA to look for SNVs, translocations, various other things. We can look at the RNA of the tumor, and then we can integrate both of those. And I'm going to just kind of give you a little bit of a flavor of what we're doing with these methodologies, since we won't have time to get into detail. So this is a plot called the circles plot that just quickly summarizes the large data set that we obtained so far. We have over 200 whole genomes that have been sequenced, over 250 RNA samples, and we can compare those to the panel assays such as the UCSF500. We have a wide variety of cancers. We're focused primarily on non-brain tumor solid tumors, but we have sequenced some brain tumors and some leukemia samples as well. And this just kind of summarizes a little bit the cohort, just to emphasize a couple of things. One, we've sequenced quite a few, and we're very proud of this. We've been able to sequence about 73 metastatic samples, which are oftentimes very difficult to obtain. Of course, we do this with consent from the patient, but even when we have consent, we have to, well, actually, you should say we, the team that we have here in the lab, sort of sits outside the operating room, works closely with our surgical colleagues to make sure we can capture as many of these samples as we can. We've also sequenced a fair number of rare tumors that have been underrepresented in prior studies. This is a similar panel to what I showed you before for the tumor mutational burden, but just to emphasize here, we're not limited to only being able to evaluate the hypermutated tumors, because since we're looking at the whole genome, we have a much larger, many thousands more mutations. So we can look at mutational signatures across the entire cohort, not just those that are hypermutated. We do notice, for example, that, and perhaps not surprising, that when we compare untreated tumors on the left versus the treated tumors on the right, so these are tumors that have been treated with some of the very intensive chemotherapy regimens that you're familiar with, us talking about, cisplatin, doxorubicin, various forms of radiation. And, when we treat these tumors, not surprisingly, we accumulate mutations in those tumors. And, what the relevance of that is to further treatment of the patient is something we're quite interested in evaluating. So, for example, here, this is looking at, again, these mutational signatures, and it's a very complicated plot, so I won't spend too much time on it, but just to draw your attention to this line here, the 6th line from the bottom, platinum drugs. So there's a specific mutational signature associated with delivery of cisplatin, and we see that very easily identifiable in the subset of patients that we know have gotten cisplatin. So, for example, osteosarcomas, non-rhabdoid soft tissue sarcomas. And there are other mutational signatures I mentioned earlier, the APOBEC signature, which I'll talk a little bit about later. With whole genome sequencing data, we can identify not only single base pair signatures, but also double base pair signatures and insertion deletion signatures. And again, this is an incredibly rich data set for evaluating these tumors and understanding sort of how they got kind of where they are. So this is another one of these case studies that we published earlier this year in collaboration with Elliot Stieglitz and his group. Anya Levinson, one of our fellows, was the first author. And this is kind of a different way of looking at this data. So when you see a patient who has a very unusual, we call exceptional response. So this is a patient that Dr. Stieglitz and Dr. Levinson saw that was kind of, had a limited repertoire of options, very advanced cervical adenocarcinoma in a young woman, a teenager. And they decided to basically kind of as a last resort to treat with immunotherapy. And patient had a very dramatic response. We could say that she's essentially cured from her disease. And so we were retrospectively asked to kind of, come in and help figure out, well, why did she have such a, exceptional response? And is this something we could use to learn, to how to identify patients who might have this exceptional response in the future? And so one of the things we noticed when we did this signature analysis for this patient is that she has this signatures 2 and 13, which are markers of this APOBEC, which is a particular mechanism of oncogenesis that leads to accumulation of alterations that could potentially be picked up by the immune system as neoepitopes. And so that suggests that maybe this is the reason why she responded. So now we've been kind of on the lookout using this molecular signature analysis mutational signature analysis, excuse me, for other patients who might respond similarly to immunotherapy. So this is just a figure kind of highlighting that we now have the gene expression signatures, because we've also done RNA sequencing for this panel, a very large panel of samples. And generally they cluster by disease, as you would expect. So the osteosarcoma shown here, for example, in 10 cluster together. The rhabdomyosarcomas, cluster together in 14. But we do see these, what we call outliers. So these are samples that kind of cluster away from other samples with the same diagnosis. And so that can sometimes give us some indication that there's something unique about the sample. And some of the work that we do in the lab is trying to kind of understand that sort of unique aspect of specific samples. We've also been able to identify, for example, a very large number of unique or sometimes very rare fusion events that could potentially have significant clinical relevance. So this is just a table that highlights again in the N-terminal fusion partner and the C-terminal fusion partner. It's a much denser figure than the one I showed you before, because again, now we're looking at RNA, not just at the DNA panel data. And we've identified a large number of very rare fusions that we think might be oncogenic drivers, and I'll give you a couple of examples of those. So one of the ways we find fusion that we think might be particularly relevant is by looking for the downstream, the 3' partner that has very high levels of expression. So if you see here, for example, in the red boxes are shown two fusions that are well known: the Fox fusion in rhabdomyosarcoma and the SSX1 fusion in synovial sarcoma. And on the left, you see the samples that have the fusion. On the right, you see the samples that do not have the fusion. And one thing you'll notice is that what we're showing here is level of expression. So the level of expression of this truncated fusion fragment is much higher in the samples that have the fusion compared to the ones that don't. So if we use that same pattern to look for kind of more novel fusions that are less well studied, and those are the ones in blue. Again, here's two examples of an RBM38 fusion and a Spark fusion. Again, the patients that have the fusion have much higher levels of expression compared to the ones that don't. So we think that those might be kind of a sort of an indicator that this fusion is important for this patient. So, for example, in this case, this is a kind of a really interesting patient who leukemia that had been treated prior with a drug called Asparaginase, which basically works to deplete asparagine in the plasma. And it turns out that most leukemias are very sensitive to this, and because they don't make their own asparagine, and the way you make asparagine is through an enzyme called ASNS, so asparagine synthase. But what we noticed in this particular patient is that they had a fusion between a gene called KMT2E and ASNS, which led to very high levels of expression of ASNS in this patient, as you can see here. So this is the kind of, again, showing the level of expression across a large cohort, and the little asterisk shows the expression in this particular patient. And so you can see that using two different cohorts, this patient has extremely high levels of expression of ASNS. And so we think that this might be the mechanism for how this patient became resistant, was by upregulating the gene through the fusion event. Here's another patient with osteosarcoma, where we don't usually see targetable alterations, but this patient, a unique patient, actually, as far as I know, unique to the literature. There's never been a patient described like this. Has a fusion between IGF-1R and NTRK3, extremely high levels of expression of the NTRK3 gene. And this patient, for reasons that I won't get into, had limited therapeutic opportunities, and so actually is currently being treated with an NTRK inhibitor. And so we'll see if that turns out to lead to a longer survival. We can also look at immunoprofiles. So in the RNAseq data, we can identify tumors that have a strong infiltration of immune subsets, which may again make them more susceptible to immunotherapy, or might help us to understand the unique aspects of their disease. So I won't get into that for lack of time. Lastly, I just wanted to mention that one of the things that, as I mentioned earlier, that we're quite interested in is these longitudinal cases where we have multiple samples from the same patient. And I wanted to just end by pointing out a couple of vignettes of individual patients that we followed over time like this. So this is the patient that we saw, one of the very, very early patients we saw when we first started this effort, who unfortunately had a Ewing sarcoma at age 9, and then for a variety of reasons, the patient was treated with local radiation to the site, and then developed over time a secondary malignancy 5 years later that was basically a radiation-induced sarcoma. So this is the case where we actually had access to the DNA of the original Ewing's tumor, and then over the course of the progression of this patient, we were able to obtain material for a patient-derived xenograft, as well as later on from a metastasis, multiple samples. Unfortunately, this patient didn't respond to additional therapy and is deceased. But it's, I think, a very illustrative example of how, as we can kind of bring this kind of information more into real time, we might be able to use it to advance the care of our patients. So this is the cohort of all of our Ewing samples that we've evaluated. The one that I'm highlighting here is the patient that we're talking about. And what's unique about this patient is they actually had a diagnosis evidence of a mismatch repair signature, which could, this is 10 years ago, so we didn't know how to look for this. But if we had known it at the time, that might have potentially impacted on the therapy that the patient would have received, and suggested that an immune checkpoint blockade could have been considered. Unfortunately, that wasn't done because we didn't know about it. The patient then went on to develop this secondary malignancy, which is difficult to see here, but if you look carefully, you'll notice that the mutational signatures overall of the secondary sarcomas are different than they were for the Ewing's tumor. And you can see that perhaps better here, comparing the Ewing's to the secondary malignancies. One thing that's very obvious is that there's now emergence of this cisplatin signature, and that's because the patient was treated with cisplatin, and you can see that kind of in the scar of what cisplatin does to a tumor when you treat it. These are the insertion deletion signatures for that same patient. We'll skip talking too much about that. But just to, again, this is a very illustrative case where we see over time emergence of multiple mutational signatures that could have perhaps impacted on the therapy of the patient, both at diagnosis and as as the tumor evolved in this particular patient. Just briefly mention a second case. This is a hepatoblastoma, where again we had access to multiple samples over the course of disease, as shown here. Again, unfortunately, this patient was deceased. But this is a hepatoblastoma where we obtained multiple samples from the original liver resection, and then also from a metastasis shown on the far right. And these are, again, the mutational signatures, and the bottom are the single base pair signatures, in the middle the double base pair, and above the indel signatures. And what you'll notice most significantly is that the metastasis has dramatically expanded the overall tumor mutational burden in this patient. We also notice that when we look at the copy number changes, that the metastasis shown on the right has a pattern of copy number change that's unique to one of the 3 initial tumors that were isolated from the liver, suggesting that one of these, the one at the top, was probably the clone that gave rise to the metastasis. So again, this is very rich data that we're very excited to further study in the lab, with ultimately the goal of, not just doing research, but figuring out how to impact on the patients in real time. So just to summarize, the DNA panel assays, I think, provide us with a significant amount of information. But again, in a lot of our cases, we need to dive deeper. So we hope that this kind of more expansive and integrative approach using whole genome and RNA sequencing will provide us with many more specific opportunities to impact on the care of our patients. So I just wanted to end by thanking the members of the molecular oncology team shown here. I wanted to highlight very much Michelle Tursky, who is the program manager, who really does a fantastic job of helping to develop this precision oncology effort here at UCSF. And then the members of my team, and highlighted in red, particularly Henry, Marcus, Alex, are the computational biologists who did all the analysis that I showed you so far. And Stan and Maria have done all the work to kind of isolate and make these samples available for sequencing. And Avanti Shah is a former member of our division who is now left to go back to UT Southwestern, but also helped do much of this work. So thank you for your time, and I'm happy to answer any questions.
Thank you very much, Alejandro, for this fantastic talk. We have a few minutes for questions, and if you could be so kind, if you have a question, to put it in the Q&A so that we can all review it, and I'll read them out loud.
Maybe I can start with a question while we're waiting for others. That was really wonderful, Alejandro. Thank you. You sort of alluded to it in your discussion of the whole genome sequencing and RNA sequencing. So maybe that answered my question. But I was just wondering, with the UCSF500, I imagine that new tumor genes are being discovered. And so is there any effort to expand that panel of genes as you to add more genes that might be relevant, especially given the finding that pediatric tumors you don't seem to get as many hits on that panel?
Yeah, no, that's a great question. And yeah, so one of the advantages of having our own assay that was developed here in-house is that, yeah, we can modify it and adapt it as we learn more. And I think they're now in version 3, and the group over in CCGL led by JP Granert and Jessica Van Ziffel, and our genomics officer for the entire UCSF campus, Alex Radkovic. They're always kind of adding new things to this and modifying it to make it more relevant, particularly adding new fusions as we learn more about these cancers. I think the point I was trying to make about pediatrics is that we can expand a DNA panel only so far, but a lot of the kind of the signal that we're looking for is not in the DNA. It's either expression changes driven by structural alterations or epigenetic changes, and so I think incorporating RNA is going to be very important. But yeah, I think the test definitely is sort of a work in progress and constantly evolving.
As I sort of expected, questions are now rolling in, so I'm not sure if we're going to get to all of them. But the first question is, thank you so much for sharing all that work with us. I was wondering if there's been any effort to tie proteomics into these sets with the hope of identifying neoantigens or tumor-specific targets on the surface of these tumor cells.
Yeah, a fantastic question. Well, in terms of neoepitopes, there are ways to do that by looking at whole genome and RNA. And that's something that we're actively trying to build out in our, sort of as a research test. I didn't really get a chance to get into it, but that one case study that I mentioned of the young woman with the cervical adenocarcinoma that had a sort of a dramatic response, we did do a neoepitope analysis in her retrospectively, and she had a very high neoepitope burden. And so we think that that's part of the reason why she responded to the immunotherapy. So that's something that we're quite interested in doing. Proteomics specifically, we have not incorporated into this, mostly because it's a bit out of our area of expertise. But I think it's definitely kind of the next frontier that should be looked at.
And then another question that I'm going to just highlight here, because I think lots of people probably want to know this, is from Dr. Matei, who's asking, how does one access this rich resource for research? What's the process, and is it open to other outside researchers?
Yeah, so the easiest way to access it up front is through the CBio portal that I mentioned, which is anybody who has a UCSF credential can access it through My Access. We just ask that you sign some conditions that you're not going to share the data or try to re-identify patients, obviously, things like that. And then once you've identified a cohort that you're particularly interested in, then we can either work with you directly, or you can go to the IRB and ask for your own access to the data independent of us. But we have lots of, kind of tools and sort of in the back end that we're happy to share with people. If you want to look at, specific aspects of the data and don't have the computational background to do it. So, the idea is really that the data should be accessible to everybody. And the reason we put all these kind of cohort slides together is just to kind of show people what's there, right? And I think it is a incredibly rich resource that that's only been, it's the tip of the iceberg is what we've looked at.
Thank you so much. I want to thank you again for this fantastic talk. I'm cognizant of the time. It's 1:01, so I don't know if you have time to stick around for just a moment for other questions.
I have time, yeah. I'm happy to answer any other questions. Sorry, I went over time.
Dr. Hirsch to finish it up for people who have to go.
Yeah, so that's great. If you're able to stay on, and people who can stay on can continue the conversation. But I want to thank Dr. Sweet-Cordero for a fabulous talk, and all of the participants, and a special welcome again to our applicants to the molecular medicine program. And I wish everybody a good rest of your week. Thank you.
Thank you, Dr. Hirsch.
Thank you, Dr. Hirsch. And next week is we're talking about COVID-19 and vaccines, so I hope lots of people will come. I think a few people are going to stick around, so let me ask a couple more questions, Alejandro. One of them is, are there any future pipeline plans to incorporate ATAC-seq or other assays of chromatin architecture, such as for the ASNS fusion case?
Yeah, for ATAC-seq. Yeah, so ATAC-seq is, for those of you not familiar with that, is a technology to look at chromosome accessibility. And it can be a very powerful way to identify not just the gene expression changes that are present in a particular sample, but to look at the sort of the potential for state changes or response to different drugs or whatever in a particular sample. And there's been some nice work done from Howard Chang's lab, kind of applying that to clinical samples. We have not done that kind of routinely. We have started using ATAC-seq kind of to look at some research samples in the lab, particularly the patient-derived xenograft samples. We are quite interested in expanding that to clinical samples. If the person who asked the question wants to follow up with me, I'd love to talk more. It's a very powerful technology and actually not terribly expensive. So I think it's definitely something we'd like to think about.
And then any thoughts about using other methods such as long-range sequencing that would pick up methylation?
Yeah, I mean, I think that there are various ways to pick up methylation. Long-range sequencing is one of them, but there are other ways to do it as well. We haven't expanded into methylation. I mean, we're limited by the limitation to this is I guess well, through twofold. One is the expense. Well, the amount of material available is oftentimes limiting. Some of these technologies require that the material be, particularly, for example, long-range sequencing, that the material be collected and processed in a very specific way. So you need more material. And then the last one is cost. And the cost is sometimes the cost of the actual sequencing, although in most cases that's come down so dramatically that it's not so much of the issue. The bigger issue is the computational cost. So the cost of people who know how to analyze this data is quite significant, but also just the the computational infrastructure required to do these analyses is not trivial. So that turns out to be our limitation. But if we're always looking for collaborators and people with interesting unique expertise, and happy to talk offline.
And then the last question asks about, do you have any suggestions of ways to help when insurance companies decline to pay for the UCSF500 panel?
Yes, well, if it's on the pediatric side, we have a pair of fantastic patient navigators who are very keen to do that. So please don't let the insurance issue be a problem on the pediatric or AYA side. On the adult side, we do struggle a little bit more, although I will say that the UCSF500 has now been approved by CMS for Medicare. So that is a huge step forward. So any Medicare patient should be eligible. Yeah, so if you're having particular issues with that, please reach out to myself or to Michelle Tursky, and we'd be happy to kind of share our tricks of the trade.
Great, thank you so much, and thank you for sticking around for a few extra minutes. Again, thanks everybody for attending, and have a good rest of your day.
Thank you.